Hey everyone, welcome to BioBusters. In this video, we'll cover everything you need to know for Subtopic 1.4 membrane structure of Topic 1, which is cell biology of the IB Biology HL curriculum. I'll begin by introducing some of the qualities of cell membranes. We'll then look at reasons for transporting substances across the cell membrane and what substances exactly are transported. We'll then take a look at the three types of passive transport: simple diffusion, facilitated diffusion, and osmosis. Then I'll explain the factors that affect the rate of diffusion. Also, we'll look at osmolarity, and finally, we'll take a look at bulk transport. Let's get started. Two of the most important qualities of cell membranes are: one, that they're semi-permeable, meaning only certain materials can freely cross. Typically, large and charged substances are blocked. And two, they're selective, meaning membrane proteins regulate the passage of material that cannot freely cross. Now, why would substances want to cross the cell membrane? Well, two of the main reasons are for cell defense, for example, killing pathogens, and for cellular homeostasis. Now, you may wonder, how do cells know which materials need to pass through? Well, those that are imported are those essential for cell metabolism, for example, glucose, hormones, ions, etc. And those that are exported include waste, toxic products, and substances that need to be secreted, such as enzymes and hormones. And finally, how do cells go about transporting these materials across the membrane? Well, they can either use a form of passive or active transport. Let's go over passive transport first. Passive transport is the movement of material along a concentration gradient from an area of high to low concentration. Because of this process does not require energy, so no ATP hydrolysis needs to occur. There are three types of passive transport: simple diffusion, facilitated diffusion, and osmosis. Simple diffusion is the movement of small and non-polar lipophilic molecules until the molecules become evenly dispersed. It occurs in a gaseous or liquid medium. Examples of molecules that use this form of transport are oxygen and carbon dioxide. Now, if the particles are too large or are charged, facilitated diffusion is used. Facilitated diffusion uses a membrane protein to transport the particles from an area of high to low concentration. So, with the aid of membrane proteins, ions and larger molecules can pass through. The two types of proteins that mediate facilitated diffusion are channel and carrier proteins. Examples of molecules that use passive transport are sucrose and potassium ions. Now let's go over the two types of proteins. Channel proteins are only used in passive transport. They regulate the passage of certain ions in response to certain stimuli via a pore. And compared to carrier proteins, they have a faster rate of transport. Carrier proteins can be used in passive or active transport. To translocate a specific solute across the membrane, the solute binds to the protein, who then undergoes a conformational change to translocate the molecule across the membrane. Let's look at factors that affect the rate of diffusion. Well, first off, temperature. The higher the temperature, the faster the rate of diffusion because the particles will have more kinetic energy. Second, we have the surface area of the membrane. Again, the larger the surface area, the faster the rate of diffusion because more molecules can diffuse at once. Third, the size of the particles. If the particles are smaller, they'll diffuse faster because larger particles experience greater resistance. And lastly, the concentration gradient. The higher the gradient, the faster the particles will move from the area of high to low concentration to reach equilibrium. And finally, osmosis. Osmosis is the movement of water molecules across a semi-permeable membrane from low to high solute concentrations until equilibrium is reached. Because solutes cannot cross a cell membrane unaided, water moves to equalize the two solutions. Osmolarity is a measure of solute concentration. It takes into account the number of osmoles of a solute per liter of solution. Solutions can be categorized three ways according to osmolarity. Keep in mind that the following terms are relative, so they're only used for comparison. Solutions with high osmolarity are called hypertonic. They have a high solute concentration, so water leaves the cell, causing it to shrink. Solutions with low osmolarity are called hypotonic. They have a low solute concentration, so water enters the cell, causing it to swell. And solutions with equal osmolarity are called isotonic. They have the same solute concentration, so there's no net flow of water, and thus the cell does not change shape. Water will always move from the hypotonic solution to the hypertonic solution. So for example, when do we use osmosis in real life? Well, for instance, in the preservation of tissues and organs in medical procedures. Tissues and organs must be kept in solution to prevent cellular desiccation, and the solution needs to have the same osmolarity as the tissue or organ, so they must be isotonic to prevent osmosis. What would happen if osmosis occurred uncontrollably in animal and plant cells? Well, in animal cells, if too much water left the cell, it would shrivel, known as crenation, and if too much water entered the cell, it would swell and maybe even burst, known as lysis. In plants, however, the results are different as the effects are moderated by the inflexible cell wall. If too much water left the cell, the cytoplasm would shrink, known as plasmolysis, but the cell wall would maintain its structured shape. And if too much water entered the cell, the cytoplasm would expand, known as turgor. However, it would be unable to rupture due to the cell wall constraints. To estimate the osmolarity of a sample, you have to bathe it in solutions with known osmolarities. When placed in hypertonic solutions, the tissue would lose water. When placed in hypotonic solutions, it would gain water. By weighing the sample before and after, we can see if the tissue lost or gained water. If there is no mass change, it means the solution is isotonic. Now, the second type of transport besides passive is active transport. It's the movement of materials against the concentration gradient, so from low to high concentration of solute. Because of this, it requires energy, so ATP hydrolysis does occur. Due to their use of energy, it involves the use of carrier proteins, also known as protein pumps. With protein pumps, the solute binds to the protein pump on one side of the membrane. ATP is hydrolyzed into ADP plus a phosphate group to create a conformational change in the protein pump. The solute is translocated across the membrane and it is finally released on the other side. There are two types of active transport. Primary active transport, which is the direct use of metabolic energy to mediate transport, and secondary or indirect active transport, which involves coupling the molecule with another one moving along a gradient. An example of active transport occurs in the roots of plants. Sometimes there is not a high enough concentration of some minerals in the soil to make them diffuse into the cells of roots. So, energy is used to move those substances in. Another example is the sodium-potassium pump, which exchanges 3 sodium ions that move out of the cell with 2 potassium ions that move into the cell. There's one last concept for this subunit, bulk transport. This occurs when cells have to secrete or absorb much larger quantities of a substance or even take up other cells. Bulk transport is capable of happening because of the membrane fluidity and flexibility, which allows for the breaking and reforming of the bilayer. So larger materials can enter and leave the cell without crossing the membrane. Because it requires ATP hydrolysis, this process is active. The two types of bulk transport are endocytosis and exocytosis. In endocytosis, large substances or bulk amounts of smaller substances enter the cell without crossing the membrane. The material to be internalized is then surrounded by an area of cell membrane and sealed to form an intracellular vesicle containing it. There are two types of endocytosis. Phagocytosis, which is the ingestion of solids, or pinocytosis, which is the ingestion of liquids or dissolved substances. If receptor proteins are used, the process becomes known as receptor-mediated endocytosis. In this process, receptor proteins on the cell surface are used to capture a specific target molecule. When receptors bind to their specific target molecule, endocytosis is triggered and the receptors and their attached molecules are taken into the cell in a vesicle. Coat proteins give the vesicle its rounded shape and help it bud off from the membrane. The other type of bulk transport is exocytosis. This occurs when large substances exit the cell without crossing the membrane. There are two types: excretion, which is when waste products are secreted, and secretion, which is when proteins made by ribosomes on the rough ER are passed to the Golgi apparatus via vesicles, are then processed and packaged and released in vesicles again that fuse with the plasma membrane for secretion outside the cell. And that's it for Subtopic 1.4 of the IB Biology curriculum. Thanks for watching!